Welcome to Hema Now, the podcast that brings you expert perspectives and the latest advancements in hematology. I'm your host, Dr. Jonathan Sakia, and today we have the pleasure of speaking with Dr. Marc Hoffman, an associate professor in hematologic malignancies and cellular therapeutics at the University of Kansas Medical Center. Dr. Hoffman's academic journey began at Yale University, where he graduated magna cum laude with a degree in molecular biophysics and biochemistry. He went on to earn his medical degree from the University of Pennsylvania, followed by a fellowship in hematology and oncology at MD Anderson Cancer Center, a world-famous institution. Today, he serves as the medical director of both lymphoma and myeloma at the University of Kansas Cancer Center. His research and clinical interests focus on lymphoma, myeloma, and hematologic malignancies, with a particular emphasis on improving patient outcomes through innovative treatments and clinical trials. He's also a key member of the Southwest Oncology Group, where he contributes to the Lymphoma Subcommittee and has been an advocate for advancing lymphoma and myeloma therapies. When he's not making strides in hematology, Dr. Hoffman enjoys playing golf, strength training, and cooking. And as any busy parent can relate, he's also found ferrying his children to their various events. Dr. Hoffman, it's a real pleasure to have you on the podcast. So without further ado, let's head over to the discussion.
All right, so welcome, Dr. Marc Hoffman.
Absolutely. It's great to be here. Thank you for inviting
Fantastic.
me.
You're most welcome. So Marc, let's start gently. You're clearly a very busy guy professionally, and I know you're into physical fitness. And I always like to ask about the other stuff before we dig into medicine. So, favorite meal to cook?
I made a duck ragu recently with some homemade gnocchi that was pretty killer. That was pretty out of the park. I can't wait to make it again. I'm happy it's getting cooler. It is a cool season dish. It's not a summer dish.
Yeah,
Kind
it
of stick
truly
to the
is.
ribs food, but I think it
Yeah.
might be our Christmas dinner. So,
If you've never tried it, cassoulet. It's a
oh yeah.
French dish made with duck and sausage and cannelli beans and wine. And what I found is you have some, you eat it. The next day, you take it out the fridge, you add some more wine. It never
Mhm.
goes away. It lives forever. It just keeps renewing itself.
Well, especially if you're adding more wine every time,
Yeah,
Jonathan.
yeah,
I
yeah.
mean,
Well,
that sort of tends to allow things to happen. The other cool thing about cassoulet, I think, is that it's better. The longer you let it rest, the better it
yeah,
gets. So 3 day old
absolutely.
cassoulet is better than 1 day old cassoulet, which is better than fresh cassoulet. So yeah.
I'm with you. I'm with you. And of course, you're just adding, fruit. Wine is just fruit. So
It's
they tell
true.
us we have to get a lot more fruit. So anyway, Mark, let's dig into some of the medicine. AI rocks because without it, I wouldn't have known what molecular biophysics is, which is what you did at undergrad. Biochemistry, I get. So according to my AI tool, essentially it explains biological processes at the molecular level by examining physical forces governing interactions and behavior, focusing on understanding how the physical properties of molecules contribute to the roles they play in biology within living organisms. First of all, did AI trick me? Is that correct? And what inspired you to do that course? And the third part of the question, how has it colored your work thus far?
So, I mean, I think in all honesty, so I went to a posh university over here in the U.S. that has trouble naming majors, and so rather than just having biology and biochemistry, we had like evolutionary and environmental biology, and then molecular, cellular, and developmental biology, and they had to create these titles that made it seem more impressive than it was. My course was basically biochemistry. I think part of the reason that they described it that way is that Tom Stites was at Yale and he solved a lot of the initial crystal structures for enzymes. And in that process, you can start to delineate what the molecular interactions are that are occurring between molecules as they're interacting and as enzymes are doing chemistry. And so that physics dictates the chemistry, which then dictates the biology. So the old adage is that all physics is math, and that all chemistry is physics, and that all biology is chemistry. And that sort of holds true as you look through the hierarchy. It's just that as you can tell, I mean, the sum is clearly greater than the parts, right? I mean, you can look at physical interactions among enzymes, which help predict the chemistry, which help predict the biology, but they don't explain how it is that an artist can create something that when you look at it, you feel a certain way. There's clearly more to the world than that. So how I got into it, to be honest with you, I was just interested in it. I wasn't trying to do anything specific. I was not thinking that I was going to be a doctor when I was an undergraduate. I liked doing physics. I liked solving the puzzles. The folks that taught the courses within that division were exceptional, and then proved to be exceptional. There were multiple Nobel Prize winners. So Jennifer Doudna, who won a Nobel Prize for CRISPR, she was one of the people that lectured me when I was an undergraduate. And so it was something that I was just sort of naturally drawn to. So it wasn't anything that was more complicated than just simply an individual following his or her intellectual interests.
Yeah, fascinating. There's a physicist over here who he was played keyboards in a rock band in the 70s or 80s, a guy named Brian Cox, and he's a superb commentator for the general public, and he does a podcast with a comedian, and he maintains that everything is physics, right? Chemistry is physics, biology is physics, and these are just subdomains of physics. Funny guy. If you've never, the show is called The Infinite Monkey Cage. Very, very different to this podcast, but well worth listening to. So, Mark, what took you into hematology and oncology? You said you didn't see yourself as moving into medicine. What took you into hem-onc, and what would you say to young doctors who were considering a career in your specialty today?
Yeah, I mean, I would give them, to be perfectly honest, with the same advice that I got. So when I talked to old seasoned physicians who weren't necessarily even people that I was training with, these were family friends and others, the universal theme was go where you like the patients. Where do you like the patient interactions? Where do you have the interest and the passion? Because at the end of the day, for most physicians, there's occasionally some people that do very hardcore basic science research who have very little patient interaction. But a decent chunk of your job is patient care. And even folks that do a lot of clinical research, it's usually at least 40% of your days. And folks that like me that are more clinical, it's 70 to 80%. So and my journey there, again, it was nothing more complicated than sort of following what I liked. I thought, and so getting sort of to answer the question you asked me previously that I didn't bother to answer. The what I studied in undergrad and those specific molecular and chemical interactions were ever present in cancer therapeutics. So all of those basic biological systems were all there. The chemistry that was being done was interesting. These were concepts I was familiar with. They were enzymes I was familiar with. And so the therapeutics were interesting. And then I really loved the interaction that you have with patients. Sometimes it can be challenging to get patients' attention when you're talking about things like hypertension and diabetes and the need to lose weight and those types of things. You see eyes glass over, and people want to have their cassoulet and add extra wine to it. When you get a cancer diagnosis, you have somebody's attention, and I liked that intensity of the human interaction. I liked participating in sort of the drama of my patients' lives. And so that bit really connected with the science. And so it all kind of came together to be a specialty that made a lot of sense. And I mean, who knows whether it was 100% the correct decision. I think I probably could have been happy doing a number of different things, but most decisions that one makes in their life become the right decisions if you went into it for the right reasons and you have the right attitude about the challenges that those positions face. So it has turned into a wonderful career for me, and I really enjoy it.
Well, we're going to dig into your career, but what would you say to the last part of my question was, what would you say to young doctors? Would you say, because I would guess it's even more exciting now than it was when you got into it. I mean, compared to when I graduated, Mark, what you guys can do, I'll give you a perspective. I've been involved for many years with a triumvirate of children's charities, Starlight, Starbright, and First Star. And I know that when I first qualified and got involved, what one could do for kids who were diagnosed with leukemias, the outlook today is dramatically different.
Yeah, there have been therapeutic revolutions in a number of diseases, and even I've been in practice, out of fellowship for now almost 10 years, and that's been long enough to see several diseases completely shift in terms of what we do therapeutically. So again, I think for young doctors, it's really a matter of following what you're passionate about. A lot of folks, especially in the US, I don't know how this works over in the UK and in Europe, but I meet a lot of younger medical students that are really worried about lifestyle choices. And I'm not here to say that managing your lifestyle and work-life balance are not important. They're critically important to your happiness. But that is work and that's life. And if you're doing work that you don't really enjoy, then your work-life balance isn't going to be particularly great either. And so having something that's, quote, a worse lifestyle, but with better work is probably going to be a more enjoyable and more manageable work-life balance than anything else. So I really encourage people to follow their passions, do things that they're interested in, certainly maintain and heavily invest in your personal wellness. But you have to do things from a work side that make you excited to go to work every day. And if you're not doing those things, this is a really hard job, right? There's a lot of things to not like about being a doctor. And so if you don't like the work can become a real problem. And so I think that it's really critical that people follow their passions, and everything else can work itself out provided that you think it's important. If you think work-life balance is important, it will be important and you'll figure out a way to make it work.
I think you said some very wise things there. The first thing is go where you like the patients. I've not heard someone say that before, but it makes eminent sense. As a surgeon, I kind of got off on the fact that someone came to me with a problem that was tangible, visible, sometimes palpable. I could fix it. And then they would leave without the problem very often, sometimes for good, something like a hernia, sometimes for at least a period of time with a colon cancer. So I get that. And I have to say that I don't know that nowadays we're doing such a good job as when you qualified, and certainly when I qualified. And I hear people talking about burnout, because for my generation, I don't think I've ever worked a day in my life. And being a surgeon is the biggest privilege of being a human being. It's going to work every day. It's fascinating. And you're right. I think you fix the other things you want to do and can still have a very rich life. So good on you for saying that. So getting a bit more granular about your work, this year you published a fascinating review on the mechanisms of immune evasion and novel treatments for relapsed and refractory diffuse B-cell lymphoma. Can you give us an overview of this element of treating this phase of B-cell lymphoma?
Yeah, I think that just to sort of put this all into a little bit of context. So we've had chemotherapy approaches for aggressive lymphomas for somewhere around 60, 65, 70 years, somewhere along those lines. And the big developments that occurred were the recognition that we needed to have combination therapy. And so CHOP was developed in the early to mid 70s. That incorporated a number of different drugs which had good efficacy against lymphomas, but also had non-overlapping toxicity profiles. So you could give all of the drugs in a cycle. And then there were some optimization studies to figure out the number of cycles that patients required. The next big advance occurred in the late 90s with the addition of rituximab and with monoclonal antibodies. And so when those drugs came out and were added to a chemotherapy backbone, we saw significant advantages in cure fractions and in overall survival among patients who had large cell lymphoma. Since that point in time, to be honest with you, up until about 5-6 years ago, where we've started to have some new stuff that's been developed, by and large, we were just playing around with chemoimmunotherapy, and there were a number of trials that were adding different drugs on, a lot of which have been negative. And so R-CHOP remains the standard of care throughout the world. In spite of having been around for 30 years, it's proven very difficult to beat. So where things are headed is that there's about a third or so of patients that are not cured with frontline chemoimmunotherapy who have large cell lymphoma. And those patients historically were managed with salvage therapy. So they got different chemoimmunotherapy combinations. And if they were fortunate enough to respond to those, they were consolidated with an autologous stem cell transplant. One of the challenges of that approach is that a number of those patients were refractory to chemotherapy, so they didn't respond to whatever you gave them in the salvage setting. The other is that a number of patients were not eligible to receive an autograft. So those patients who were a little bit too old or infirm or who had other comorbidities were frequently not eligible to undergo a curative intent autograft procedure. So where things have headed over the course of the last 7 or 8 years in large cell in the relapse setting has been the advent of a couple of different therapeutic modalities. So the first is CAR T cells. And in CAR T cells, what we're doing by and large, this is the normal standard of care autologous CAR T cell, and we can talk about other more novel constructs if you're interested. But we're actually harvesting the patient's own T cells. And as you are probably aware, the majority of T cells, cytotoxic CD8 positive T cells, were designed to kill off virally infected cells. And so the external piece of those cells in the T cell receptor identifies peptides. Well, it turns out that using those peptide identifications, you can kill off some cancers, but it's a little bit too specific. And so what these CAR T cells do is that they actually chop off the T cell receptor, maintain the signaling apparatus that allows the cell to remain cytotoxic. But on top of it, and using the CRISPR technology, they actually add in an FAB fragment of a monoclonal antibody that can then go in and recognize a specific target. And the currently FDA-approved constructs are CD19-directed autologous CAR T cells. There have been a couple of randomized clinical trials showing superiority in cure fractions and survival in patients who receive CAR T cells in second line compared to intent to autograft. That piece is one section. The other section is actually using the patient's own T cells, so what we call endogenous T cells, and activating those through bispecific monoclonal antibodies. So these compounds bind to cancer cells, but also bind to T cells and bring them into close proximity and lead to some degree of immune activation. So those compounds, as well as CAR T cells, appear to be resulting. The data on the bispecifics is a little bit less mature, but there are probably some patients who are achieving durable long-term remissions and potentially cured with those therapies. There definitively are in patients who receive CAR T cells. So those are better, right? They're better than what we were doing before, but we still have a lot of those patients. In fact, the majority of patients who receive both of those types of therapies still end up relapsing. And the question is why? And so what this paper was looking into was just reviewing all of the different ways in which the lymphoma cells can evade immune surveillance, number one. So just what your body is doing naturally. And then what happens to patients when they're exposed to drug therapy and why it is that they eventually will end up failing. And some of it has to do with T cell fitness. Some of it has to do with potentially antigen loss. And so there's a number of different theories about why this is occurring. But at the end of the day, the immune system is simply, it's activating sometimes, but then it's not remaining activated for long enough to result in durable disease control. And so that's what that paper was looking into. One of our very excellent senior residents, along with one of my fellows, did most of the work for it. And I was there mostly in a supervisory capacity. But that's what that paper was looking at.
That's fascinating. You were also involved in the Bruin study, investigating new treatment approaches for B-cell malignancies. Can you explain the goals of Bruin and what kind of impact it's going to have?
Yeah, so I think Bruin is one of the cooler phase 1 studies that's ever been done. I think it may be the only phase 1 study I've ever seen that recruited 300 patients. So there's a class of molecules called BTK inhibitors. Okay, so there is a B cell receptor that sits on the outside of most B as in boy cells. And it turns out that tonic signaling through the B cell receptor apparatus is involved as an oncogenic pathway in a number of different types of blood cancers. And so when I say an oncogenic pathway, basically these are getting turned on, these genes are getting turned on, and that's leading to unrestricted growth among the cells. So BTK inhibitors have fundamentally altered the therapeutic landscape in a number of diseases, probably the most important of which is CLL, but it also has made significant advances in mantle cell lymphoma, marginal zone lymphoma. There was a recent approval in follicular lymphoma, Waldenstrom macroglobulinemia, and in a number of other rarer diseases in which we often will use them off-label. So all of the initial drugs that were developed had a couple of limitations. One was that the toxicity profile. They included a couple of somewhat irritating toxicities, which among later generations of the drugs have gotten better. The second is that there was therapeutic resistance. And so all of these were what we call a covalent BTK inhibitor, meaning that the enzyme was physically binding to the drug. So the drug binds specifically to the enzyme, creates a covalent bond, is physically attached to it, and leads to degradation and death of the enzyme. When this area that it's binding, the binding site, is mutated, the drug can no longer bind, and there's some resistance in that cell line, and those cells can still grow. So what the Bruin study was looking at was a non-covalent BTK inhibitor called pirtobrutinib, and pirtobrutinib used allosteric binding, so it came in and bound into a pocket rather than actually physically binding to the cell. And when it bound, it led to degradation and death of the enzyme. And so you got the same therapeutic result, but it was able to overcome some of those mutationally driven events. Interestingly, in all of the diseases where it does appear that it's mutations that are driving progression events, so predominantly CLL, but it occurs in several other malignancies, these drugs work extraordinarily well. So pirtobrutinib was able to rescue the vast majority of patients. It's an unbelievable waterfall plot. If you look at the study, there were almost no patients that didn't respond to therapy.
Wow.
Among those diseases like mantle cell lymphoma, where it's probably not mutationally driven, it's driven by the development of alternative oncogenic pathways, some NF-kappa-B, some other stuff in mantle cell lymphoma, the responses were not quite as brilliant. So there were patients who responded and responded quite well, but there were a number of patients who did not. And so that waterfall plot was a little bit more sobering. But the cool part about it is not only did it work, but there's basically almost no toxicity. So giving pirtobrutinib is probably one of the single easiest drugs I've ever given for cancer therapy in my life. It basically doesn't cause anything. So the atrial fibrillation that we saw with the covalent BTK inhibitors, by and large, it doesn't cause. There's not any significant diarrhea. There still are some sort of weird infectious things that you have to worry about, particularly if you're giving it with high-dose steroids. But those are quite uncommon. And then I had the only patient that came off the study was my patient who had a significant grade 3 rash and had to come off. But he's the only person I've seen that's developed a rash. So it's a really cool compound, and the reason that they enrolled so many patients is they kept seeing it working in the early phase studies, and the efficacy signals were so dramatic that they kept opening more and more expansion cohorts. And so we enrolled, hundreds and hundreds of patients on that study, and so it's been a very big success. Pirtobrutinib is now in a randomized phase 3 study against ibrutinib in treatment-naive CLL. And recently, we're about to see the results from a positive study, which is with pirtobrutinib plus venetoclax and rituximab compared to venetoclax and rituximab in refractory CLL. So the drug's really moving forward. It has activity in Richter syndrome, which is something that we have always had challenges treating. So it's a really cool drug, and it was fun to be a part of the development of it.
What sort of timescale before this could be in the clinic?
It's available in the U.S. As it's FDA approved, I think, for third line CLL. But I give it off label for a variety of different things. And since we have a whole set of national guidelines called the NCCN guidelines that you may or may not be familiar with. But if something's listed as an option there, generally we can get insurance coverage for it. And I think it was recognized among the authors of those guidelines that this drug has an important role in the therapy of a variety of different diseases, even though we don't have phase 3 studies. And to be honest, we're probably not going to get large randomized phase 3 studies in a disease like Waldenstrom's. There's just not enough patients. There's not enough willpower. Recruiting that many patients, by the time you recruit the patients and watch them, it's a question of how relevant the study question is.
Sure, absolutely. I have a friend with this condition. I'll have to check and see if it's available here and if it's being given. And just as an aside, I'd heard that Waldenstrom had been cancelled and that it's now been renamed. I might have misheard.
I don't know. If you know that, you may be
I
more
don't
in the
know.
know. You're
My
more in the press, Jonathan. I'm not as savvy with following up on the newspapers.
Yeah, my grasp on reality is so tenuous these days. But yeah,
What you're offering, though, is a trick that the infectious disease doctors usually do. They usually, every 10 years, they rename bacteria so that we have to
I
consult them. Because I'm like,
know.
what the hell is this?
know. I
I don't know what this is.
can't keep up with it. Yeah, I'm sure I had a conversation with someone, and I actually knew what Waldenstrom's macroglobulinemia was, which is pretty good going because surgeons don't normally use that many syllables in any word. We just speak grunt. But yeah, but I was told, I'll have to fact check that. So, changing subject a little bit, just slightly, diffuse large B-cell lymphoma is the most common form of non-Hodgkin's, I believe, and patients who relapse or don't respond to initial treatment face potentially grim prospects. And you've mentioned CAR T offering hope by reprogramming patients' own T cells to target cancer cells, but there are barriers which hinder CAR T therapy deployment access. Can you talk to us about some of the impediments and how we might address them? And some of them, of course, the things like cost, access.
Yeah, I mean, I think it's a really good question, and Jonathan, I have worked on some panels in the U.S. on this, but also worked with some folks in Spain, and it was interesting. In the U.S. we have this concept that our insurance apparatus, where we don't have a public safety net insurance program, is responsible for the vast majority of issues with drug and therapy access. And there's a little bit of truth to that for sure. I'm not here to say that there's not a component of that. But it was interesting how many factors we shared with the Spanish physicians regarding other things that are keeping people from getting CAR T cells, which I would argue actually outnumber the people that are uninsured. So the one of the challenges we have with CAR T cells is that patients generally need to travel somewhere. So you have a person who is sick, who's relapsing with lymphoma. They're going to have to travel to another center. Often they will need to be there for a prolonged period of time. And after they receive their CAR infusion, they need to be there for a minimum of 14 days. And currently we're doing 28. We actually were just publishing a study. In fact, I think it got published last week, looking at a potential 14-day stay to try and reduce that burden on the amount of time that they're away from home. Even if this is just an hour and a half away, it's a big enough space that getting to the center, having a caregiver that's available, somebody that can be with you 24/7 during that time frame, is a lot to ask. And so that's a really big barrier. A lot of people are very reluctant to ask family members or friends to participate, even though those folks may be willing to step up. They see this as being an insurmountable obstacle. So a lot of it boils down to distance, caregiver needs. Occasionally, there's some biological factors where access to pheresis slots, meaning where we actually pull the cells out, can be limited. Occasionally, there's some insurance denials, but the vast majority of the time, it's really just human factors. And so things that people have thought about are, I think one of the pieces here is us on the academic side working better with community doctors to establish clearer referral networks. And I think the biggest impact that folks in the community they can have is providing a very strident recommendation for CAR T cells. So the stronger that they feel and tell the patient that they need CAR T, the more likely they are to be able to overcome some of these barriers. We also need to figure out ways to transfer some of that care back and provide clearer and more consistent algorithms to community doctors who are ready to take these patients back and continue to care for them. Figure out ways to reduce the amount of time that these folks need caregivers. figuring out more outpatient strategies for delivery of CAR T. There's a lot that can be done, but most of these are human problems. And as you're aware, typically the biological problems are easier to solve than the human ones. The human ones frequently require a little bit more work. Do you build some sort of hotel type situation where you can house these patients? Those exist at a variety of centers throughout the world. I know Fred Hutch has a big one in Seattle, since they're responsible for caring for patients in Alaska and Montana and Idaho, where they're traveling long distances to get there. Um, and so that a lot of, but it's these are really human factors that we need to figure out ways to overcome, and we can adjust some of the therapies, but a lot of it's more social factors in those senses.
Yeah, and I think these are global social factors. And the
Hmm.
word access is sort of all-encompassing. There can be many reasons why people have access issues, and it can be simply that they're hundreds of miles away from the center. It can be insurance coverage, availability of drug. I mean, I'm being told that there's shortages of the GLP-1 drugs. I was in LA. I worked in the States for many years, and I was in LA not too long ago, having lunch with a dear old chum, and he was telling me that as far as he knew, everyone in Beverly Hills was taking GLP-1s,
Yeah.
whether they need it or not. But of course, it has impact on access for those people who are morbidly obese or have diabetes. So change of tack slightly. This could be an interesting one. So you serve and have previously served on several international advisory boards for a variety of conditions. They're very often supported by pharmaceutical companies that obviously have a dog in the race. So let me state my bias up front. I've been outspoken in condemning the pharma scolds. These are people who think that the pharmaceutical industry shouldn't be talking to us, right? We shouldn't have the life science industry involved in these discussions. So I've already said, I think this is nonsense. What would your perspective be? Because I let me just firstly also say, after many, many years in medicine, is it possible that there are doctors who behave badly? Yep. Is it possible there are pharmaceutical companies who behave badly? Yep, absolutely. Do the vast majority? Not at all. The vast majority of doctors are caring, diligent, ethical, committed people who really want to do the right thing for their patients. So have at it, Mark.
Yeah, I mean, I think it's a really interesting question, and it gets at a lot of challenges that you have when you're in an industry where there's a very limited number of people who actually understand what's going on. Okay. So when I start talking about covalent BTK inhibitors and some of these other things, there's huge chunks of that I don't understand, that I'm dependent upon basic scientists to help me interpret. That group gets even smaller. I mean, there's certain aspects of really in deep cancer biology where very few people know what's actually happening. And those people are not all in one space, right? They are mixed into a variety of different places.
Yeah.
Those people have to talk. If we're going to solve problems, those folks have to talk. And I think that the view that pharmaceutical companies and the healthcare professions need to be separate is a naive one. And I think it's naive for a few different reasons. The first is somebody has to take on the risk of turning an idea into a product, right? So at some point, somebody has to take this idea that you have, develop a drug for it, take on the risk of doing a study of that drug, prove that that drug works, and then sell it. And that risk is the only people that are in a position to be able to take that on in any reasonable way is the pharmaceutical industry. Somebody has to do that. They're really good at it. So their ability to take one drug and figure out how to change very small aspects of it, they do that masterfully well, way better than an academic lab ever could. So they're taking on that risk. That's one piece. The second piece is when we interact with pharma companies, my interactions with them are extremely frank. I will tell them if they have a drug that I think is bad, and that I think they shouldn't develop it in certain conditions. I will tell them the limitations of the way that they've done studies. The other piece is that it gives you an opportunity to influence the way that studies are designed, right? At the end of the day, we want clinical trials that are designed in order to benefit patients. So, not just benefit future patients, but benefit the patients who are actually on the clinical trial, where at the very least, they're not going to be harmed. And you're hoping that you can design a study where at least some of those patients you're actually helping. Okay. If they don't have feedback from us as investigators who have done a lot of these clinical trials, they are prone to developing studies that are not therapeutically relevant. There have been multiple studies that we have been presented in these types of advisory boards, and I've said, you can't do that study. That's an unethical study. No one should enroll on it. I would recommend you don't do that. And those studies don't end up moving forward, right? I don't see them again as iterations. So that feedback ends up being incredibly important. So I think that now, that all being said, you have to figure out a way, how do you mitigate these conflicts of interest? And clearly, many years ago, decades ago, when the industry was a lot less regulated, there were major problems. You had somebody that came in, they showed you how many prescriptions of whatever drug you had written. And if you wrote a certain number, they took you out on a really nice golf trip to Ireland and all expenses paid, and took your entire family and never had to report it. And you didn't have to report it as a physician. That's clearly not the right way that things should be done. The question is, has the pendulum swung a little bit too far? I don't know. I think that's kind of hard, but it is very highly regulated. You can't, the sales group of pharma companies and the science group of pharma companies, I can't even be in the same room with both of them at the same time. They can introduce one another and then they have to leave. Somebody has to leave in order for us to have a conversation. So I think the tight regulation in some sense is good and is necessary, but I think it's an incredibly naive point of view to suggest that we are not going to have the drugs that you want in order to treat your cancers unless we are facilitated and have a way to work together to design the drugs themselves and then design the landmark clinical trials that'll prove that they work so that they'll be available for everyone.
Yeah, I'm with you. And I think it's important that even some of our colleagues do not appreciate what it costs. And I've been involved in developing a number of medical technologies and a couple of drugs. It costs a bloody fortune and it takes forever. And the regulations to protect, against, the bad effects of something, there's all sorts of checks and oversights for everything. I know more about devices than I do about drug development, but I just wish that there could be an honest debate and we could take the politics and the vitriol out of it, because otherwise, these marvelous new therapeutics that you have access to and that, we're all going to be patients sooner or later, we won't have those. So going back to, I guess, my prior question about involvement with various groups, you chair the Lymphoma Myeloma Disease Working Group, a group of investigators, research folks involved in lymphoma and myeloma trials. Can you tell us about your work in this entity and I guess how it differs from some of the other things you do?
Yeah, so that's it's something called a DWG, and what we do is that we're responsible, and this is true of all centers that perform clinical research. We're responsible for choosing a portfolio of clinical trials that we think we can enroll on, right? So that's always a question: do we have the patient population where we feel like we can put patients on the study? And then the second piece is that study a relevant study? Is it going to advance the science of the disease? Is it going to make a difference to patients? If we have the results of that study, if we knew those results today, would we be treating patients differently? And so we have, as all situations are, we have limited resources. I don't have as many clinical research coordinators as I want. I only have so much clinic space. There's only so many regulatory people. And so we have debates in that context in our monthly meetings, sometimes which can be quite robust, about which trials we should open and why we should open them. And so that's the forum in which that takes place. I'm also on something called a protocol review and monitoring committee, which does a couple of different oversight functions. One of which is we review all of the new protocols and determine whether those are protocols and ask mainly enrollment questions. Can we enroll? but also asks some really critical questions about whether the study questions that are in there are valid. And then we monitor every year each of these trials to make sure that they're meeting accrual goals and meeting scientific goals. And that group actually has the power to stop trials. So we can actually discontinue and remove a trial from our portfolio. And so it's nice to be on both of those, where I'm on the activating side and the deactivating side. The deactivating side is really helpful when we're trying to pick studies, and I'm saying, well, this study is not going to go anywhere. But I think it's probably one of the more interesting jobs that I do, because I learned so much about how my colleagues think about these diseases, and we get to go through so many different protocols and really understand how is the protocol written? Is that going to make this problematic to enroll on? And sometimes we're able to actually provide feedback to companies, and they adjust their protocols in relationship to the feedback we've provided, to be able to make it a better study. So that's the work that we do through that committee.
That's fantastic. So as we head towards the end, Mark, what is at or just over the horizon in the areas of hemonc that you work in that have you most excited?
Yeah, I think there's multiple pieces to this, so I could go a ton of different directions. I'll take two examples of things that I think are really important. One is that in multiple diseases, and I think CLL is really leading the charge in this, we're coming up with ways of treating patients where they're receiving highly effective, non-chemotherapy targeted drugs in combination for a fixed duration that's resulting in incredibly deep remissions and potentially curing some of those patients. But it is taking a set of therapies, we're getting rid of chemotherapy completely and trying to get off of the indefinite therapy plan. So it's a pretty remarkable revolution, considering that only 12 years ago we were mainly giving chemoimmunotherapy to just about everybody with that disease. So that's one massive development status. And then the second piece is sort of optimizing these immune therapies. So between CAR T cells and bispecifics, this is a little bit hard to explain without some slides, but there are ways to make in vivo CAR T cells using multiple different antibodies that are getting looked at, which would certainly be a lot easier than having to do pheresis and autologous CAR T cells. And then there's optimization of the CAR itself, right? So this is the old joke that currently we're driving the old Ford Model T and we need to be having a Mustang, right? So the current car that we have is a little bit bulky. It's got some limitations. There's clearly things about it that can be better. And we're in the process of revising and defining that. And so I think it's really advancement of multi-agent targeted therapy and optimization of immune therapy are, I think, the two big groups of drug development that are really moving forward. And I think will set the tone for the next 3 to 5 years of development in lymphoid malignancies.
Yeah, well, as I said earlier in our conversation, Mark, that to an outside observer, the changes in your specialty have been awesome. They have in mine as well. The technology that we have at our disposal and the things that we can do for patients and the results that we can achieve. I always remember the old expression, 5 years after you leave medical school, 50% of what you learn is either incorrect or irrelevant. The problem is knowing which 50%. And I qualified so damn long ago, I don't think anything was still bloody relevant. So I always
it's like the Cellular process, right? There's still got to be at least 2%, right?
Yeah, that's right. Eventually, hopefully. And sometimes things circle back round. I love the whole neuromodulation space. You go back to Victorian patented medicines or patented advertisements, and they were advertising electrical shock generators to treat everything like Parkinson's disease. And lo and behold, that's what we do nowadays, right? Yeah. So fascinating stuff. So I always like to ask people a bit of a fanciful question. If you stumble across a genie and you're granted 3 wishes for your area of healthcare, what would you wish for other than the perfect cassoulet recipe?
So the first, and I actually think this is probably the most important, because I think it would make physician and patient lives so much easier. If we could get simple and universal drug coverage for all of our patients without having to go through the headaches of insurance approvals and the misfortune of some patients who are uninsured just simply not being able to have access for drugs, that's a heartbreaking situation. So I would rub the genie and ask her for that. I think the second piece is we could go through some therapeutic advances and understanding those, but I think some of it would be addressing again some of those social factors that we talked about with CAR T access. That we could figure out a way to solve some of those so that it was easier to get patients caregivers. We could more than double the number of people who are cured with large cell lymphoma just by getting them in for CAR T cells, right? We don't have to do anything. We don't need to develop new ones. They don't need to be better. They just need to show up at our door. And so I think optimizing that process from a social angle and simplifying it. And then I think that the last piece is figuring out a way that we can continue to have the important drug development that we do and the incredibly rapid pace of new therapies, better tolerated therapies that are easier to take. Can we do that and also not have those costs of those therapies break the bank? As of right now, the way everything is structured, the large profit motivation is really what's leading to the development of these drugs. And if that goes away, it's not clear how much would be developed in this space. And so figuring out solutions to how we develop those problems. Those big
Right.
structural issues, the biology will figure itself out if we figure out those things.
Yeah, I think you're absolutely right. Well, I'm afraid that's all we've got time for today. My guess is the speed at which things are happening, we should have you back on again, and you can update us, and we can start with a recipe exchange. So, Dr. Marc Hoffman, really want to thank you for taking the time to be with us today and for everything that you're doing for patients, and sharing it on this episode of Hema Now is hugely valued. Real pleasure chatting with you.
Absolutely, Jonathan. I really appreciate the opportunity to come on and look forward to talking to you again.
Well, I hope you enjoyed that episode of Hema Now, our bi-weekly podcast on hematology. Please consider subscribing on either Apple Podcasts or Spotify, or frankly, wherever you get your podcasts, so that you never miss an episode. Like us on social media, tell your friends and colleagues, and please check out the archives because they're growing if you've missed any episodes. And please join us again in 2 weeks for another episode. Until then, I'm Dr. Jonathan Sackia. I'm proud to be your host. Please, everyone, stay safe, stay well, stay curious. Bye for now.